are in the second session of Mall Bio. So, today we are going to deal specially with DNA packaging, followed by the search for genetic material, experiments involved in that. After that, we will be looking into the RNA world, followed by DNA replication. Of course, in our previous class, I have dealt about central dogma of molecular biology, right? Both in prokaryotes and eukaryotes along with locations, right now beta? Now, if you see the genetic material that is present in prokaryotes, fine. In prokaryotes, of course, the DNA is not completely scattered into the cytoplasm. Okay, DNA is not completely scattered in the cytoplasm, but it is at a particular place, right? It is holding at a particular place because there are some positively charged proteins binding to it. DNA is negatively charged, we know. Okay, so those positively charged proteins which are present in prokaryotes are not histone proteins. They are not histone proteins. They are non-histone polyamines, non-histone proteins which are known as polyamines. Okay, they will bind to the negatively charged DNA and it will allot it to a particular place in the cytoplasm. Fine, right? Now, this is addressed as nucleoid or genophore. Nucleoid or genophore. Clear? Okay. Now, if you see in eukaryotes, here in prokaryotes it is double stranded circular DNA. In eukaryotes it will be double stranded linear DNA, okay, wherein the negatively charged DNA is attached or aligned with the positively charged histone proteins. Okay, here positively charged histone proteins are present. Histone proteins are positively charged because they have basic amino acids like lysine and arginine. Fine. So, if you see first how this chromosome is packed into the complete condensed structure. First supercoiled DNA, chromatin fibers it forms into, then DNA and histone proteins will come into picture. So, first process of packaging is what? Supercoiled form of DNA, followed by chromatin thread like structure, and then comes DNA along with histone proteins. DNA along with histone proteins is nucleosome model, which is the highlighted segment in our syllabus, nucleosome model, and that gives you the double helical structure of DNA. Fine, you have understood the steps of DNA packaging. In prokaryotes there are no histone proteins, it has non-histone proteins which are polyamines. In eukaryotes positively charged histone proteins are present, they are positively charged because they have lysine and arginine. Here you see the nucleosome model. So, the negatively charged DNA here, negatively charged DNA is winded up to positively charged histone proteins. Fine, right? Negatively charged DNA is winded up to positively charged histone proteins. These histone proteins are positively charged, okay, because they have lysine and arginine into it. One complete DNA is wrapped into a histone octamer. What is this? Histone octamer. Histone octamer, which has 8 histone proteins. Fine, right, beta, 8 histone proteins. You have 2 sets of 8 histone proteins. Total number of histone proteins are 8 histone proteins. How many types? 4 types of histone proteins are present. Remember the statements carefully. You will not get confused in statement based questions. Total number of histone proteins are 8. How many types? 4. two sets of 4 types are present. What are they? H2A, H2B, H3, H4. Two sets of this are present. Fine, right? Which forms a histone octamer around which the DNA is wrapped. Now there is a question. How much amount of DNA is wrapped around the histone octamer? Okay. DNA length is measured in the form of base pairs, number of base pairs. It is 200 base pairs that is wrapping around one histone octamer. How many base pairs of DNA beta? 200 base pairs of DNA is wrapped around 200 base pairs of DNA is wrapped around one histone octamer. This forms a new body. Fine, right? So in one nucleosome, how much amount of DNA is present? 200 base pair. Now from one DNA it is connecting to other again, right? It forms another histone octamer. Fine. This DNA is addressed as what? Linker DNA. Okay. Linker DNA is binded up between two histone octamers. Okay. This is linker DNA. Fine, right? Linker DNA which is wrapping around two histone octamers. Fine. So this linker DNA which is wrapping around two histone octamers is holded by H1 histone proteins. How much amount of DNA is present in linker DNA? It is 80 base pairs of DNA which is present in the linker DNA. 80 base pairs of DNA is present in the linker DNA. Clear? So here can you see 200 base pairs of DNA, new body it is called as, which is wrapping in one complete histone octamer. The DNA which is moving from one histone octamer to other, that connecting DNA is addressed as what ma? Linker DNA. What will be the length of linker DNA? It will be 80 base pairs. Clear till here completely? Now this is addressed as nucleosome model. As the compaction is increasing, as the packaging of DNA is further increasing, what happens is non-histone proteins comes into picture. Non-histone chromosomal proteins. Okay, those you will call it as non-histone chromosomal proteins. Okay, non-histone chromosomal proteins can be scaffold or structural proteins, structural non-histone chromosomal proteins. It can be regulatory non-histone chromosomal proteins. Regulatory non-histone chromosomal proteins includes the enzymes like DNA polymerases, and RNA polymerases. Fine. This is about non-histone chromosomal proteins which increases further compaction of DNA. So you are clear with new body? How much amount of DNA is present in one nucleosome? Single nucleosome, 200 base pairs. In linker DNA, 80 base pairs. So in a single nucleosome, how much turn it will complete? One DNA it will complete 1/3/4 of turn. Okay, it completes how much turn? 1/3/4 turn is completed in one new body. That minute segment is linker DNA which finishes off the complete turn. Clear? Clear with histone proteins? Clear with nucleosome model? Now, after compaction of the DNA, what happens is you will come across two types of DNA segments. Okay, the regions where DNA is highly packed, is addressed as what? Heterochromatin region. This is the region which is highly packed DNA. Complete compaction of DNA is seen. Okay. The regions where the nucleosomes is loosely packed, here you can see they are loosely packed, right? This is euchromatin region. As such, after compaction of the DNA, okay, after condensation of DNA, you can say in proper terms, you will come across two regions. One is euchromatin and the second is heterochromatin region. Euchromatin and heterochromatin. Remember, euchromatin is transcriptionally active. Okay, euchromatin is loosely packed and it is transcriptionally active. Heterochromatin is tightly packed and it is transcriptionally inactive. Fine, right? About euchromatin and heterochromatin, you are clear? Now, search for genetic material. Now, scientists have begun in the late 18th century, okay, and the beginning of 19th century. They are starting up the process of what is that transforming across the generations. Okay, of course, Mendel has addressed it in the form of factors, but what is that actually transferring? Nobody knows. Okay, then the search for genetic material has begun. First experiment is what? Transforming principle. First and foremost experiment is transforming principle, which is given by Frederick Griffith. And what is the model organism over here? Model organism is Streptococcus pneumonia. Fine, right? It is Streptococcus pneumonia. This is the model organism. You have two strains of Streptococcus pneumonia. Okay, one is S strain and the other is R strain. S strain is addressed as smooth strain, which is virulent. Why is it virulent? If you consider this as a bacterial cell, around the bacterial cell it has a smooth shiny coat. It has a smooth shiny polysaccharide coat, which is giving it an extra coating and extra support to survive in the host immune system. Okay, this remains unaffected in the host body. Okay, when this is unaffected in the host body, this causes the death of the host, right? So this S strain is what beta? Virulent strain, because it has a smooth shiny coat which is polysaccharide coat. Okay, it has smooth shiny polysaccharide coat. R strain, you see, it is a rough strain. Okay, it doesn't have any smooth coat over here or around it. It is a rough strain like this, doesn't have any coat over it. Okay, so this rough strain bacteria is non-virulent. It cannot survive in the host body because it doesn't have any extra coating over it, right? No polysaccharide coat over it. So this bacteria is non-virulent. It will die in the host body. Clear with two strains of Streptococcus pneumonia? One is R strain and second is S strain. Fine, right? Now you have two strains. 4 models are given. In the first phase, what happens is the living S strain is injected into the mice. When living S strain is injected into the mice, mice dies, of course, because this is virulent and this can survive. Fine. Second is R strain, which is virulent or non-virulent? Non-virulent. When it is non-virulent, the mice survives or it is alive. Third model, what happens is the S strain will be heat killed. Okay, S strain bacteria is heat killed. When it is heat killed, S strain bacteria is injected into the mice. Mice is surviving because due to heating the S strain, what happens is the S strain polysaccharide coat is removed off. There is no coat over it now. Now this is non-virulent and the mice survives. Now at the fourth step, what happens is living R strain is mixed with the heat killed S strain. Living R strain is mixed with heat killed S strain. When this is added up, what happens is there is something that is being transferred from the dead S strain to living R strain, which is making the R strain virulent. Okay, this is making R strain virulent. But what is that is actually transferring, nobody found out. Okay, Griffith has not said what is that actually transferred from dead S strain to living R strain, which is making R strain virulent, that is not explained. But he said that there is a transforming principle, some material is being transferred from heat killed S strain to R strain, so that R strain has synthesized polysaccharide coat over it, polysaccharide coat over it and became virulent. Fine, you are clear with the 4 steps, right, of search for genetic material? Now, biochemical characterization of transforming principle. Biochemical characterization of transforming principle. Now you have got an idea that something is being transferred so that R strain become virulent, but you have to identify, right, what is that actually transferred. So that experiment is done, which is addressed as biochemical characterization of transforming principle. For this, what happened is they have extracted the pure biochemical from that, what, from the mixture of heat killed S strain and R strain mixture is there, right. From that pure biochemical is extracted. Pure biochemical is extracted from that, okay, where into 3 test tubes the strain is being taken. The strain is being taken into 3 test tubes. What happened is, into one of the test tube, proteinase is added. Proteinase will digest what? Proteins, okay, and that is injected into the mice. Mice is surviving. S cells are appearing in this. So this is not affecting any transformation. When proteinase is added, only proteins are being degenerated and that is not affecting the process of transformation. So it is not protein. Cleared, right? Second, we have a doubt on RNA. We will check it out. So here what happened in the second test tube, RNase is added. Okay, RNase is added. And this is digesting what? RNA. This is also not affecting the transformation. S cells can be even found in this. Even after an RNase is added, you can see the S cells. Fine, right? So RNA is not the transforming principle here. RNA is not that is transforming. Now our last step, that is DNase. When DNase is added, DNA got degenerated. Here after that, if you see, S cells are totally absent here. There are no S cells. Okay. So it is what that is transforming from heat killed S strain to R strain? DNA. Okay, don't laugh, this is a huge process that is being taken place in the history and you are laughing at it, huh? Okay, so this is DNA, fine. DNA that is being transferred from the heat killed S strain to R strain is finally categorized by biochemical characterization by what scientists? Avery, Oswald Avery, MacLeod and MC Carte in 1944. Fine, right? Yeah. So you are clear with this experiment, right? Even statement based questions you should be confidently answering it. Okay. Now, biochemical characterization, people are not satisfied. Now we need an experimental proof for that also. Okay. So an unequivocal proof was given by Hershey and Chase experiment. These three experiments beta you have to remember. First is what? Griffith's transforming principle. Second is what? Biochemical characterization. Third is what? Unequivocal proof by Hershey and Chase experiment. These three are the standard examples that you have to remember with respect to the discovery of genetic material. Fine, these three experiments. In sequence you have to remember. Go through the NCERT lines properly, statement based questions you will work out easily. Fine. Now, what is this Hershey and Chase experiment? They have worked out on T2 bacteriophage. What is the model organism over here? T2 bacteriophage. T2 bacteriophage, if you see, T2 bacteriophage, you know bacteriophage is what? Which is infecting bacteria and it has a genetic material and protein coat, right? So the genetic material consists of phosphoric acid. Each nucleotide is a combination of phosphate, nitrogen base and sugar. So phosphate is present in the genetic material. One radioactive labeled phosphate is added to the DNA. What is the radioactive label? P32. Fine. Same ways, the radioactive isotope is also given to the protein coat. Protein coat has what? Amino acids. Sulfur containing amino acids are present on the protein coat and the sulfur radioactive label is what? S35. Two such bacteriophages are taken. One is labeled at its DNA, other is labeled at its protein coat. Fine. Protein coat labeling is what ma? S35. Genetic material labeling is what? P32. Clear, right? Radioactive labeling. Now what happens is three steps will be in this experiment. One is infection, that bacteriophage infecting the bacteria. Second is blending and third is centrifugation. Fine. So the infection step, what happens? We know that even we have learnt it in lytic and lysogenic cycles, wherein the genetic material from the bacteriophage is injected into the bacteria. Now second step is what? Blending. Mixing of the genetic material, both viral genetic material and bacterial genetic material. Now viral genetic material is integrated into the bacterial genetic material, yes or no? Yeah. After centrifugation, what is centrifugation? That in a test tube, the mixture is taken after blending, the bacterial mixture is taken. Now it is analyzed whether the protein coat that is entering into the bacteria or genetic material, that is DNA, is being entered into the bacteria. That is our check line. Okay. So here after centrifugation, if you see, after centrifugation we know at the base retentate will be formed, dense material will be at the base and supernatant will be at the top. Supernatant particles will be at the top, which are light in weight, right? So if you compare genetic material and protein coat, genetic material is dense, right? DNA is dense because it has whole lot of particles like sugar, phosphoric acid and nitrogen bases, okay? Whereas protein coat just has amino acids. It is just of what? Amino acid sequence. So when it is being checked at the retentate or at the base, the genetic material is being identified. First label, that is with S35, which is over the protein coat. Protein coat, of course, they will be at the top in the supernatant, okay. No radioactive S35 is detected in the cells. In the bacterial cells, no radioactive isotope S35 labelings are detected. Only radioactive detected in the supernatant of the centrifugation tubes. Fine, right? Next, if you see radioactive P32, okay, these are detected inside the cells. No radioactive is detected on the supernatant. Do you have any doubts here in infection, blending and centrifugation? Clear, right? Any doubts anywhere, ma? Yeah, enough. Hershey and Chase experiment is clear to you, no? Now it is the time to know the clear differences between DNA and RNA. What are the key differences? And then we will move on to the RNA world. So in the search of genetic material, we have clearly analyzed how the genetic material is discussed. Two segments are clear to us. One is how DNA packing is done, what is DNA packaging and how is it done in the eukaryotes, and what is the procedure in prokaryotes. Both things are clear to us. Second is the discovery of genetic material. 3 experiments are clear to us. Now, the sequential differences between DNA and RNA. First you see the occurrence of genetic material. Occurrence of genetic material. DNA acts as genetic material in most of them, even in bacteria and in eukaryotic cells. RNA also acts as genetic material in few viruses, like tobacco mosaic virus has what? Single stranded RNA. And one more example is Hubibacteriophas. Second is chemical stability. DNA is chemically and structurally stable. Okay. RNA is less stable when compared to DNA because it has free OH group at second carbon of the ribose sugar. Okay. If you can see that pentose sugar, ribose sugar, okay, C1, C2, C3, C4 and C5, 5 carbons, right. At second carbon there is free OH group. Okay, in the complete nucleotide chain if you see, the ribose sugar second carbon has free OH group. That free OH group will make the RNA reactive and unstable. Okay, that is making it what? Reactive and unstable. So, less stable, RNA is less stable because it has free OH group. It has free OH group in the ribose sugar, making it what? More liable and degradable. This makes it more liable and degradable. Fine. Next is replication ability. Fine. This can generate its own replica efficiently, which is by what? Self-replication. Whereas here, this can replicate only in the presence of some specific enzymes. Okay. Only in the presence of specific enzymes, this can replicate less efficiently. Fine. Next, mutation rate. Mutation rate is fast in RNA when compared to DNA. Mutation rate is fast in RNA because these are reactive. Next is what? Expression of characters. Expression of characters. This expresses itself through mRNA, provides stable inheritance of Mendelian characters. But here in RNA, this can directly code for protein synthesis, yes or no, in RNA viruses, and this easily expresses its characters. Next, sugar component. Deoxyribose sugar has no free OH group, whereas RNA has free OH group at second position. Okay. Take down the differences quickly. Not the complete sentences, but the gist of the points. Once it is done, let me know. Make it fast. Any doubt? Done? Now, few more differences now. Nitrogen base. We know what purines both in RNA and DNA are common. Adenine and guanine are present. But when it comes to pyrimidines, what are present in DNA? Cytosine and thymine are present in DNA. Uracil and cytosine are present in RNA. So, thymine in the place of uracil is giving extra stability to DNA. Okay. When I am dealing with nitrogen bases, I gave you the synonyms of each nitrogen base. Fine, right? So, here thymine is also known as 5-methyl uracil. Okay, 5-methyl uracil. Thymine is also addressed as 5-methyl uracil. And here this contains uracil instead of thymine will reduce the stability in RNA. Next, stability across the life cycle. Okay. These are stable across different stages, age and physiological changes, whereas RNA is unstable, we know. Next, overall stability, DNA is more stable, whereas RNA is more liable and it is easily degradable. Next, example, if you see, DNA is found in all the higher organisms, whereas RNA is the genetic material for some viruses only which are easily mutated. Okay. Example is TMB and QB bacteriophage. Take down. Just few differences about nitrogen base you take down from this slide, that is enough. Only about nitrogen base and the example. Done, ma? Now, RNA world, very important. Prior to this, you see, is it DNA or RNA that is first observed? RNA is the first genetic material. Why? I will tell you, see. Okay, RNA is the first genetic material. I will be explaining you this, I will be telling you the reasons, then we will go with the theory. Okay, RNA can act as a catalyst it plays a structural role, and it can act as an adapter. First, we know all the catalysts are proteins only mostly, okay, mostly all are proteins, but some RNA will also act as catalysts, like for example ribozymes. Example, ribozymes. Fine, right? For example, if you see ribozymes, they are of catalytic function. Example, SNRPs, which helps in splicing. Ribonuclease P, which helps in cleavage. Fine, right? Next is peptidyl transferase. This helps in formation of peptide bond. These are some catalytic functions of ribozymes. Okay, one is SNRPs, which helps in splicing. Second is ribonuclease P. Third is peptidyl transferase, which helps in the peptide bond formation. Fine. So here we go. RNA is the first genetic material which is believed to predate DNA and proteins in early life forms like some viruses only. Essential life processes such as metabolism, translation and splicing are thought to have evolved around the RNA only. These are the three essential processes like metabolism. Metabolism means biochemical reactions that are happening. Many of the biochemical reactions for which the catalysts are what? Ribozymes. Here you can see SNRPs, ribonuclease P and peptidyl transferase. Fine. So it is helpful in the biochemical reactions, in catalytic function of biochemical reactions. Next is in translation. Okay, I told you that it also acts as an adapter molecule. tRNA is an adapter, right? That is adapting what? An amino acid and synthesizing an amino acid here. Okay, that is adapting the codon of the mRNA and synthesizing an amino acid, which is acting like what? Adapter molecule. Okay, so in translation, tRNA, adapter RNA will be helpful. And in SNRPs and in splicing, SNRPs are helpful. SNRPs are also RNAs. So it does the essential functions. All these are what? Essential functions. These essential functions are revolving around what? RNA only. That is one major point to confidently say that RNA is the first genetic material. Okay, to get convinced with the statement that RNA is the first genetic material. Next, RNA served for the dual functions. What are the dual functions? One is it is acting like a genetic material and also as a catalyst. That is the reason you have to know RNA can have two forms. One is genetic RNA, second is non-genetic RNA. Genetic RNA is present in the virus, okay, like QB bacteriophage and TMB. Non-genetic RNA is present in SNRPs, in the form of SNRPs, ribonuclease and peptidyl transferases. Fine, right? So here ribozymes or catalytic RNA molecules, they will perform important biochemical reactions. Two such examples, ribonuclease P for cleavage, peptidyl transferase for formation of peptide bond. Peptidyl transferase is synthesized from 23S rRNA or 28S rRNA. RNA catalytic property is made it reactive and unstable. Okay, RNA catalytic property is making it what? Reactive and unstable, limiting its long term role as what? Hereditary material. Clear till here? Any doubts anywhere, ma? Yeah. Now, genetic code. Genetic code we will look into the next class. After this we have to move through replication. So, we are completed with what concepts? DNA packaging. Second is 3 experimental proofs for the search of genetic material, Griffith's transforming principle, biochemical characterization. Third is Hershey and Chase experiment, which is unequivocal proof that DNA is the genetic material. And then we have looked into the RNA world, okay, both genetic and non-genetic RNA. 3 concepts are clear. Now comes the major concept of mol bio, okay. The major concept is what? First, replication. We are done with central dogma, so you have to go through replication. Experimental proof of replication process, that is Michelson-Stahl's experiment. Very important for your NEET and MCERT both. Enzymatic machinery of replication is highly important. Second is transcription. Followed by post-transcriptional changes in eukaryotes. Fine. Now third, before moving on to translation, you should have a clear idea on tRNA, that is adapter RNA, okay, and genetic code. Fourth will be genetic code. When you are clear with these 2 concepts, then you can easily move on to the concept of translation. Lastly comes what? Gene expression. These concepts we will be dealing in the next class, which is major and very important. You can easily expect 2 to 3 questionnaire from genetics from this area only, your NEET or MCERT. Okay, 2 to 3 questionnaire we can expect from this domain itself of your mol bio. Fine. After gene expression, it is DNA fingerprinting and human genome project, where direct NCERT liners will be asked. We will go through it along with the NCERT. Okay. So, this will be our next class. Till here you revise and come for the next class. Fine, right? Next class is very important.